now I'm so happy today because I'm here. I can see a lot of friends and also to introduce my good friend, Mario Rivas. Mario is an extraordinary young researcher, and today we talk about RNA world. Uh, Mario, thank you so much for accepting my invitation. Please share your screen and go ahead.
Perfect, Fabio. Thank you so much. Let me share my screen. I think I will share the whole thing because I don't see the PowerPoint. But then we go. Can you see my PowerPoint slides?
Yep.
Yes.
All right. Let's go into full mode.
Hey.
Perfect. Then I would like to start by thanking Savio. He's a very good friend of mine since I was still an undergrad. And I think before he joined NoRCEL, we met in Mexico City a long time ago. And that's the main reason I am here, because he asked me to do this. And I was like, of course, my friend, this is a great opportunity for us to be like networking and everything. And before I start, I would like to do some disclaimers. First of all, opinions and the whole story that I'm gonna tell here, it's my own interpretation of events and many other stuff that I did while I was doing my postdoc. And of course, some of my colleagues in the different centers, like I used to be in Fox Lab for 4 years, and as part of the whole network with NASA, I used to be part of the Center of the Origin of Life. Many of my colleagues there will agree with me, but some of them will have their own questions about the story that I'm about to tell. Second disclaimer would be that I think I covered it all. Well, then let's start. Today I will present a whole historical point of view of the RNA world. Of course, I will include at the end some part of my own work and the work that many people have developed, because this is not possible without the collaboration of many people. First of all, I would like to start with the basis. The people that study the origin of life have the goal to find out what happened on Earth between the formation of the planet, you see, with a very harsh environment, with high temperatures, without oceans, and how we transition from that planet into a planet full of life. This is a slide with mainly eukaryotes, but of course we know that the majority of the biosphere is microbes, and those are really, really important to shape what we see today in Earth. But traditionally, there have been two ways to approach to the origin of life question. One is called the bottom up, which means how the chemistry that predates the origin of life shape all the molecules and form all the compounds that were necessary for creating life. And the top-down approach, it is by making biology comparisons, biochemistry comparisons. We try to extrapolate what would be the original state in which life originated. Most of them mainly touch one big part that is called the last common ancestor. And from there, we try to extrapolate what were the original compounds, conditions that were taking place at the origin of life. But as I will go to this talk, we will see that although the RNA world, which is the main topic of my talk, is mostly shaped in the top-down approach, the bottom-up has also been contributing in the last years to this perspective on what the RNA world is. And before I go any further, I would like to start with the basics, which means what's the RNA structure, just to benefit the whole audience, because I know there's a very broad audience with different backgrounds. So by starting, I will point out two main differences that are textbooks difference between DNA and RNA. DNA is considered a double helix, while RNA is a single helix. But we will see throughout of my talk that this is not the case. RNA is a very complex molecule that can form very different shapes, sometimes double helixes, but sometimes other shapes that are extremely complex and very difficult to form if you go with a molecule, a different polymer like DNA. The other difference is the use of the last base. The RNA, let me just use the laser. The RNA uses uracil as one of its 4 bases, while DNA uses thymine. But as you can see, the structures here, the only difference is this methyl group here. And chemically speaking, it is relevant, but it is not as relevant as we will see other chemical groups. The more relevant difference that I can talk about is the 2' OH group that is placed in the ribose between DNA and RNA. RNA has an OH, while DNA has an H. And this is considered a smallest chemical group, but it is the most important one. In several of my work have been focusing on this difference. And today I will talk about its catalytic properties, but it also has structural properties that I'll probably will discuss in a different talk. Well, over the years, the RNA molecule started as just a mere intermediary in the flux of information between DNA, which stores the information, and the proteins that are the effectors. You can recognize this scheme is based on the Francis Crick proposal of the central dogma for the molecular biology, where it states that the DNA transfers information into an RNA, which is the intermediary, and then RNA transfers information into protein in a process that is called first transcription, DNA to RNA, and RNA to protein is called translation. But DNA also replicates. That's necessary for bacteria and other organisms to duplicate its genetic material before dividing. RNA can replicate itself by an RNA polymerase, RNA dependent. We can have a very common, well-known virus flux of information, which is ribose transcription, in which you use RNA as template and you produce DNA. And of course, there are other possibilities that have not been described in any other organism today, which is reverse translation. This is probably not existent, and protein replication, which is also probably not existent. But this is the common scenario that we have in history, RNA being just the intermediary in this flux. But this intermediary turned out to be very powerful. By using the information of the 16S, the small subunit of the ribosome, and comparing that 16S among different organisms, these two men, Carl Woese and George Fox, were able to produce the first universal tree of life, which englobes eukaryotes, bacteria, and archaea. Before they used this genetic marker, most of the phylogenetic relationships between organisms were based on morphology. And of course, it's very easy to compare, for example, eukaryotes among mammals, because we have 4 extremities and a head and several organisms and bones. But what about bacteria? It's a single cell. Through morphology, we cannot compare, but through this genetic material, by comparing the sequences of the genes, we are able to derive these relationships. And this is one of the main reasons why genetic information is so important. And in this case, RNA can be carrying information that gives us a broad spectrum and a broad perspective of what life is. But today, we recognize that the role of RNA is much, much bigger than we thought. We have RNAs that regulate other genes, which are called riboswitches or microRNAs. Other RNAs that intervene in the translation process, like tRNAs, which I will talk about them a little bit later. And we have modified ribonucleotides, which are now coenzymes, amino acids like histidine, alarmons, or even antibiotics. Using the ribonucleotides, one enzyme can convert those ribonucleotides and deoxyribonucleotides to create DNA. And of course, the main star of my talk will be the ribosomal RNA, which is a large non-coding RNA which has a structure. And let's start with that part. In the RNA world, I will try to give you a small story of how we came into this idea. First of all, we are celebrating 70 years this year, 2023. We're celebrating 70 years of the discovery of the model of the DNA done by Francis Crick, James Watson, and some other people like Rosalind Franklin's data and Maurice Wilkins. But these two minds, after finishing their main goal, which was discovering the structure, of course, these creative minds do not sit and just stay around getting profits from their discoveries, but they try to increase the knowledge. And by doing so, they created this. This is part of the club, the RNA. It is called the RNA tie club because they use ties with an RNA molecule, or what is a representation of what they envision as an RNA molecule. Here you have Francis Crick, Leslie Orgel, James Watson, Alexander Rich, 4 of the founding members of the RNA tie club. But the RNA tie club has more members, right? Each of these members had the assignment of trying to elucidate the code of the transition from RNA into proteins. And to do that, they try to decipher the code going one amino acid at a time, and each amino acid was assigned to each of their members. For example, glycine here has been assigned to Richard Feynman. Melvin Cowley had histidine. You have Erwin Chergap here, the Leslie Orgel, Max Delbruck, the guy from the this virus from bacteria, and even Sidney Brenner, Francis Crick. You see all these members, and they were assigned with very different titles. For example, the synthesizer was George Gamow, the optimist was called James Watson, and the pessimist, of course, is Francis Crick. But by studying all these properties of the RNA, several of these members actually came into an idea, which I call an educated guess, which is essentially a hypothesis. Alexander Rich in 1962, Carl Woese in 1967, Leslie Orgel and Francis Crick wrote wonderful papers. I truly recommend to read those papers in 1968 about their speculations on RNA. RNA for them is, of course, an informational molecule because it has the properties of DNA, so it can contain information, and that information is passed through the ribosome into the proteins. But they have the suspicion that because of the RNA structure, RNA can have a catalytic activity. This activity at the time was not confirmed, but their experience working with RNA led them to this assumption. And the assumption was even bigger. They said that RNA can have an heterocatalytic property and an autocatalytic property. Heterocatalytic is merely being an enzyme-like structure or entity that can catalyze a chemical reaction. An autocatalytic property is being able to produce copies of itself. So it is able to produce more RNA. It was not until the early 1980s that these two people, Tom Cech and Sidney Ullman, described different ribozymes, meaning that RNA can have catalytic activity. This is the first time that we have proved that RNA has a catalytic activity. Tom Cech, working in the University of Colorado, working with the group 1 intron, which is an self-splicing intron that uses RNA's properties. The 2' OH attacks 2 times, makes a nucleophilic attacks in 2 different times by editing this group of introns. And of course, being the 2' OH, it is intrinsic that the RNA is the one that is creating this chemical reaction, the cutting and pasting of this group of introns. And Sidney Ullman, by its own in Yale University, he and his group were studying the RNase-P, which has, of course, an RNA and a part of protein. This is a big molecule that is in charge of tRNA maturation. And he and his team discovered that the catalytic part of this complex was not protein, but RNA. With this in mind, in 1986, Walter Gerber proposed the RNA world. He coined the term RNA by assuming that the RNA can work both as a genetic information and as a catalytic molecule. Well, that's the very early beginnings of the whole story of RNA. Now we will go into some of the evidence that came to support the idea of the RNA world, but that were not intended to support the RNA world hypothesis. I would like to present you the ribosome. This is a crystallographic representation of a whole ribosome from Thermus thermophilus. In gray, you can see the RNA from the big subunit. In cyan, you can see the RNA from the small subunit. The proteins are portrayed for the small subunit in blue, dark blue, and in magenta, in the large subunit. You have a tRNA molecule that is portrayed in orange, and a nascent peptide, which is still attached to this tRNA, and this peptide is going through the exit tunnel in the ribosome, where it starts folding. This image gives us very detailed information of what this big machinery from the cells is doing. You can also see the messenger RNA in yellow. This is the intermediary molecule that is carrying the information for the whole process to take place. But this is possible thanks to the work of these 3 people, Venki Ramakrishnan, Tom Stites, and Ada Jonah, which were awarded with the Nobel in Chemistry in 2009, because they were the first to crystallize such a huge complex like the ribosome. Because crystallography is very difficult, but it becomes a very tough problem when you go into bigger structures. If you start with small molecules, you can nowadays you can do it more or less easily. But if you go into bigger structures with such complexity, it became a very tough problem. But these 3 guys were able to create the first structures. Thanks to these crystallographic structures, then the first thing that people realize is that the ribosome is in fact a ribozyme, which means the catalytic center where the peptidyl transferase reaction is taking place is made out of RNA, exclusively RNA. The proteins are very far apart in terms of molecular scales. They are not even close to the PTC region. Therefore, the whole reaction is taking place in a very small pocket made entirely of RNA. And because the peptidyl transferase reaction and the PTC center, which is this pocket where the whole reaction is taking place, it's so important and it is so crucial. It is assumed that this is the ancient or the most ancient part of the whole ribosomal structure. And the first idea on how to trace the evolution of the ribosome came from the group of Lauren Williams in Georgia Tech. This image that I present to you is the image that summarized the onion model, which means that the point of origin, the center of this onion, is where the PTC is placed. And as you go out and away from that point, you go into more recent layers of RNA that were added later in the evolution. Also, with the comparison of crystallographic structures from the three domains, these guys were able to see which parts are more recent, which parts are more ancient, and derive a core, the basic core that most likely was present in the last common ancestor. In the panel A, B, and C, you have different RNAs from the small molecule, from the small subunit, sorry. In red, and in blue, you have the large subunit. And of course, the gray parts are new additions into this structure, and the common parts are in bright red for the small subunit and in blue for the large subunit. As you can see, eukaryotes have bigger parts that are not really present in the common core, so they most likely recent. And also in Lauren Williams' group, and which Lauren Williams is this guy here, and Anton Petrov were leading a group that actually came with a model that describes the ribosome evolution. In this case, the large subunit evolution. They describe 6 phases in which this ribosomal structure start growing at different times. They start with phase 1 and 2, which are, of course, in the PTC region. This that I'm pointing, this is the secondary structure of the large subunit that represents the RNA that forms the peptidyl transferase center. But that model is based on one idea. The idea is accretion. And what is accretion? Accretion is, try to see it like how ancient cultures built pyramids. Instead of building one big pyramid in a single step, they started with a small pyramid, and then they add another pyramid on top of that one, and on top of that one, they add a new one until they reach the size of the pyramid that they desire or that they could get. So most of the temples have these layers in most of the pyramids, at least in Mesoamerica, that were the way they build these pyramids. So that's accretion. It's building on top of the thing that you already have. It's like the growing of the trees in this example. Instead of letting branches to grow from the center, you have other small branches growing out of branches that are already there. And from the center of the tree, you can expand into the top of the tree by adding more branches. But you'll never create another center of the tree. You grow on what it's already there. That's the accretion model. And the molecular example is this. This is a helix, a very small helix in E. coli. When you compare that helix with, for example, anarchia, haloarchylomaris mortui, and you overlap or superimpose these structures, you see that the helix is still there, but you see other material of RNA that has been growing in the archaea. But if you go even further and compare that same helix with Saccharomyces, Saccharomyces still retains that helix. But if you superimpose this helix, you will see that apart from the material that has been growing in haloarchylomaris mortui, other part of the material were also growing in the Saccharomyces. And you can do that for Drosophila melanogaster, which is a more morphologically complex organism. You will see other regions that have grown, but the one thing remains. These starting helixes are there because this is only one example, but Lauren Williams' group has several examples of this that were the guiding force for their model. So they assume there are several signatures that allow you to see where one growth in certain organisms is taking place compared to another organism. So by comparing these structures, they were able to create this model of how the ribosome evolved. Another feature that arrived from a different group, this is Ada Jonas' group, that was derived from the crystallographic structures, was that in the PTC area, there's what they call a pseudo-symmetrical region. You see this point, red point here, represents the void of one pore where there are two pseudo-symmetrical RNA segments that hybridize together and create this pore where the PTC reaction is taking place. And that's portrayed in the whole structure of the large subunit. You can see here these two small RNA fragments, one in green and one in blue. And this proposal was done almost 20 years ago. Recently, Oxtran and Jonath were like trying to derive what are the residues that delineate that pore. But by doing so, they also realized there are other pores similar in size, but they are not catalytic. So Dr. Fox at that time was asking what is the difference between the PTC that is catalyzing the peptidyl transferase reaction and other pores that are just there in the structure but are not catalyzing anything. This is a work that I did in 2012, 2013 with George Fox's group, where we focus our attention on that question. And we arrived to this conclusion. In panel A and B, you can see that the PTC, the light of this pore is delineated by the bases. The bases are exposed into the solvent and are able to actually coordinate the RNAs and the amino acids for the peptidyl transferase reaction to take place. Compared to other pores similar in size, the phosphate backbone is the one that is facing the light of this pore. So no bases here. The backbone is the thing that is creating this pore. So chemically speaking, these two natures are the ones that are present in all the pores of RNA, because we not only look for them in RNA structures from the ribosome, but from other ribosomes and other RNA structures. So in this sense, the PTC is unique because it was the only pore that we see that all these bases are facing into the solvent and are able to coordinate tRNAs and amino acids. So in that sense, PTC is unique. Then further, we try to visit again the first proposal of the pseudo-symmetrical region to try to extrapolate the areas where these two fragments of RNA, one in green, one in blue, that I showed you earlier, that was proposed in 2003, 2005, that were the ancestors of the whole RNA from the large subunit. So we characterize all these regions where these two structures meet from different crystal structures from the three kingdoms. So we were taking advantage of the availability of RNA, of these ribosomal RNA molecules that are present in current databases to make a comparison and try to see which are the places that interact in most of these structures. Then further, we came to the realization that most of these structures are very magnesium dependent, and propose a mechanism that in the early times attach together these two fragments, the blue and the green one, into a single RNA chain by using the reaction catalyzed by one of these magnesium ions that is present in most RNA molecules. With that in mind, we recently wrote a review where we focus our attention on the PTC of the ribosome, and we describe all these characteristics, talk about the impact of having magnesium available and these non-standard interactions that we see where these two things meet together. But that was all theoretical, because many groups have been trying to synthesize one of these structures to try to see if it still catalyzes the peptidyl transferase reaction. But last year, in 2021 and 2022, these two papers came out from Ada Jonath's group, where her group were able to create not one, but 5 catalytic structures. This is the people that did all the work. This is Ada Jonath. The first author of both papers is Tanaya Bose. The person that actually proposed the pseudo-symmetrical region was Ilana Atman, and Anat Bashan has been in Ada Jonath's group and developing this theory for several years now. And this is a table that summarizes the result. As you can see in green, they have 5 different constructs from different organisms and with different sizes that were able to dimerize into a catalytic unit that was able to create a peptide bond. They have other constructs that were able to dimerize, but were not able to produce any peptide bond. And finally, they proposed several structures. Probably they did more than they report here, but they report at least 3 that were not able to dimerize nor create a peptide bond. But this is the first time somebody is reporting what they call now a proto-ribosome that is functional and it's able to create a peptide bond. So we decided to go into the details of this structure following their report, and we were able to produce a new paper based on their sequences and folding properties that we propose were responsible for a successful hybridization and a successful produce of the peptide bond. And this is the secondary structures of the old proposal in panel A, and in panel B, this is their construct based on the Thermos Thermophilus secondary structure. And in this panel, you can see the biggest construct that they have, and it is really important to pay attention to something. They only use for this construct the P region. The P region that has been in green in the previous mentions that I did, blue was taken out of the picture, and they allow two P regions to hybridize together. This construct was successful, and it is one construct that was able to hybridize, and they create a smaller construct. As you can see, as I transition from one to another, you see helix 89, if you pay attention to that one here, it's becoming shorter. And that did not affect its capability, because this construct called TTP1C is also able to produce a peptide bond. But from this construct that is also producing a peptide bond to a second construct derived from a different organism, if you pay attention to helix 74 now, you will see that the shape is changing. That shape is critical, because with this change, this construct is able to hybridize, but is not able to produce any peptide bond. So helix 74 happens to be crucial for this hybridization and for the correct production of a peptide bond. We even model the original peptidyl transferase center in panel A, and in panel B, you can see a successful construct, how it would like two P regions hybridizing together. The pore still has light, the pore is still able to create a peptide bond. But as many of the people that are familiar with the RNA world proposal, RNA is a very robust molecule in terms of its functionality, because it can be an information polymer and a catalytic entity. But it is also part of its properties that it quickly hydrolyzes itself, so it is very unstable, and there has not been any report of long chains of RNA that were able to be produced under prebiotic conditions until this paper came last year. This is a paper that was produced by Elisa Biondi and Steve Benner, two very nice guys from the community, the Origin of Life community, but very dedicated scientists. They assume several aspects or several scenarios for the production of RNA long chains, long polymers, and they realized that there's a way to produce 100 to 300 nucleotides in length of these polymers of RNA. So these small molecules that I show you with the RNA's work were around 71 to 67 in length polymers. So we're within the numbers. So in this scenario, these RNA molecules could exist in the RNA world or in the early years. So we think with these new results, we're just transitioning from a pure RNA world into RNA protein world. Why? Because most of these polymers that could be produced by this protoribosome could be helping to stabilize RNA and allow the growth of RNA molecules. And as RNA molecules grow, also the peptides grow. So this is a fragment from a very recent paper that we just published that summarizes all this information, and I'm going to read it to you, but you can also have a screenshot of this fragment, because it's really, really important, and it is the message that we want to send. We sustain that the robust scenario with a holistic view will never portray RNA evolving as an isolated molecule, but rather a molecule that can coexist and sometimes find benefit from the interactions with non-ribosome related small protopeptides of dead C peptides. Dead C peptides are summarized equally with protopeptides in these 3 papers from Moran, Frankel, Pinter, also with the group of Lauren Williams. I recommend those papers to you if you want to go deep into that one. Unlike any other RNA molecules, the ribosome constructs presented by Jonat's group also exhibit a strong magnesium requirement, because in the reports from Tanaya Bose, they realized that if they lower the concentration of magnesium, these constructs were not able to dimerize or to produce any peptide bonds. All this information adds another layer of complexity to the interactions that a protoribosome must have gone through the dawn of life. So together, divalent cations, protopeptides or deoxypeptides, and RNA must have interdependencies that ultimately drove the protoribosome's evolution. So if this theory is actually correct, and the protoribosome exists at some point, it must be interacting with some metal ions and other peptides that are also produced by other factors in the RNA world. And I cannot finish my talk without saying that in this same group, there's people like my friend Savio that has been publishing papers about the role of the transfer RNAs and proto-tRNAs in the formation of this PTC. I strongly recommend to read those papers. I myself have been reading them. I would like to present some tRNA, which is being envisioned as the ancestor of the protoribosome, and one P region extracted from Thermus thermophilus that resembles and has this structure. We'll go into these comparisons, and that would be probably the subject for another talk, but it is pretty promising that we can continue with this work. And finally, but very important, is that there's a very recent paper, actually from 4 days ago, that arrived in Nature, suggesting that RNA could be co-evolving with other peptides, with other molecules like peptide change, the small peptide change, and that cohabiting of these 2 molecules could enhance the fit of each molecule. I will strongly recommend you, if you are interested in this topic, to just go and download this paper. It's open access, so you can see another perspective of RNA together with peptides, small peptides, in an early air environment. And with that, I would like to thank you for listening, and to thank Dr. Josh Fox, Wayne Tran, Madam Tirumalai, that have been part of all the work that I have presented today. Thank you.
Thank you very much, Mario.